Okay, so I'm actually speaking as part of another life project, a kind of sister life project. It's written very small, but you can see it down here, 𝗵𝗵𝗀𝗀𝗀𝗀 2020. It's kind of a more general conservation project. It's not specifically for orchids, but I'm doing a part of that on orchids. Now, I've actually talked about this in the past. I've talked to the Life Orchids project about the work that we're doing at the moment. And if this will let me go ahead, okay. So I'm actually not going to talk about our current work today because I've already talked to you about this. This is in Italian, but you can go onto the website of Life Orchids or go onto YouTube and you can look up I Giovidi di Life Orchids, in which I explain our current activities in the 𝗵𝗵𝗀𝗀𝗀𝗀 project. What I want to do today, because this is a training course, so it's more of an applied course to understand the problems and the solutions for producing orchids. So what we'll be doing is looking at the different stages in production, the different problems, and then our experience of getting over these problems. So this is just a representation of what we're going to do. We're going to go from the seeds, or actually before that, from pollination to plants that we then put into the wild. So I will talk about translocation in this presentation, but mainly what I'll talk about are the methods to get us to that point and then to actually do the translocation successfully. So the first thing that I want to talk about is actually something that we were talking about, that Thomas talked about in the last talk, and that we then talked about in the discussion, which is pollination, the importance of pollinators in the habitat, the importance then of also using pollination as a method to maximize fruiting and the production of seeds. So this is both a good conservation technique, so producing more fruits, producing more seeds for the plants in the wild. But it's also something that's fundamental to us. If we want to produce the plants in the laboratory, we have to have seeds. And if we go and take all of the seeds that are there for these, some of them are quite rare species. So if we take all of the seeds for the laboratory, we're actually damaging the population in the wild. So the first activity that we've always done, and I think this is really a very effective technique, is to pollinate, to understand how the plants are pollinated in nature, to understand the reproductive ecology, and to use this to produce seeds. And in fact, I should just say, based on what we were saying in the discussion of the last talk, I think that basically there are species for which producing adult plants using all of the techniques that we'll see now is very effective. And there are plants for which I'm getting more and more pessimistic about the use of laboratory techniques. And I think that it's actually much more effective simply to do the pollination, to take a group of students into the field and to pollinate these plants. So you can leave a lot of seeds in situ for future generations and the natural establishment. So I think for some species, and again, with orchids, you have to be very species specific, okay? You have to think about the particular species you're working with. For some species, I've kind of, I've become more pessimistic about using laboratory techniques, whereas others, it seems to be very effective. Okay, so let's keep that in mind. The first thing to do for pollinating orchids is to understand the reproductive ecology. And in fact, the technique that we use is a technique that Charles Darwin used when he was trying to understand the pollination mechanisms. So he looked, for example, at these groups of pollen called pollinia. There are 2 pollinia in each flower, which are basically packets of wax with the pollen grains embedded in the wax. And the idea is that all of the pollen goes from one flower to another in a very, very precise, specific way. These plants produce hundreds of thousands of seeds. Each seed requires a pollen grain. Okay, each seed represents an ovule that's being fertilized by a pollen grain. So it's clear that to produce hundreds of thousands of seeds for this wind dispersal mechanism, you need hundreds of thousands of pollen grains. So this is basically, yes, sorry, Mariangela is saying the other talk that I did was in Italian, but if you want to access the other talk, you can see the photos and the videos, and you should understand some of the work that we're doing at the moment. Anyway, sorry, coming back to this. The reproductive mechanism then requires hundreds of thousands of grains of pollen grains, and orchids do this by having these packets, these pollinia. So the idea is to transfer pollinia between the flowers. And Darwin studied this using needles, actually, so steel needles, which he poked into the flower to get the pollinia. And this is basically the mechanism that we use, the method that we use. So again, Darwin was interested in the biology, understanding the biology and the reproductive ecology, but this is what we actually use to increase the amount of fruits and seeds that we have. That, you know, we can then leave most of those fruits in the wild, and we've already done something very worthwhile for the conservation of the plants in situ. Okay, and then when we take some of those fruits and some of those seeds, we know that we're not damaging the population. We've already improved the situation by pollinating and producing more fruits and seeds. So this is the method that we use. To do it, you have to understand where to get the pollen from and where to put it. So pollen is basically held in these pollinia, which are enclosed. There's a kind of covering which has to be removed. And you can stick something in there. Darwin used a needle, but you could use a pencil or a toothpick. In fact, we use toothpicks quite often. And then when you have that pollen, you have to put it in the right place. And the right place are the stigmatic surfaces, the female fertile parts that we can see. I don't know if you see the laser pointer, so I'm just going to put that. Okay, so the female stigmatic surfaces, this is where the pollen has to go. So to do this properly, you have to understand where to get the pollen from and where to put it. And once you've done that, you have more fruits and seeds produced. This is just to illustrate that you can't always do this. This is an example using a species of Gymnodenia. This is Gymnodenia 𝗵𝗶𝘀. This is Gymnodenia 𝗺𝗺𝗁𝗁𝗁𝗁. The old name is Nigritella 𝗺𝗺𝗁𝗁𝗁𝗁. But the point is that with extremely small flowered species, if you use a needle like Darwin did, you'll just destroy the flowers. This is terrible. This was a horrible experiment. It just destroyed flowers. Nowadays, we use wooden toothpicks, or you can literally simply get the stalk of a grass from nearby, and it's very effective. You can do it with that, and it doesn't damage the flowers. So there are some plants for which we find this method to be very, very effective. Here we can see some tin foil, because you can also take the pollen, wrap it up in tin foil, maybe have a cool bag, and then take that to another population and actually do a cross with another population. In the case of some orchids, particularly 𝗀𝗵𝗶𝘀𝗀𝗋, which is a kind of more primitive genus, it doesn't have two pollinia which come off the plant. It has two stamens which stay in position. And basically, you have to rip these off with some metal tweezers and transport those. It's a different pollination mechanism, which relies on insects rubbing across these stamens and taking a powder of pollen grains with them. So you do have to understand the pollination mechanism for each species in order to do this. One of the main problems that we have then is time, in the sense that very often we meet photographers who go out into the field. They take photographs of the flowers. We found the plant, we found 𝗃𝗏𝗏𝗀𝗏 rubra, fantastic, we know where it is, we know how many plants there are, I have my photographs, fine. But when we're collecting seeds for this work that we do in the laboratory, very often we have to wait for an extra month and a half or 2 months, and everything happens in those 2 months. This is literally an example of 16 𝗃𝗏𝗏𝗀𝗏 rubra plants, a very small population. And we did this hand pollination, and we came back after a month and a half, and we see plants that are dry. We see really literally where the plants were. We see that somebody had a bonfire. This disturbed ground is literally where these plants were, and the farmer, a local farmer, had stored wood on this site in the meantime. Between the flowering and when we should collect the seeds, he stored wood and then dragged it off the site. So actually one of the main problems in collecting seeds is the length of time that it takes for the maturation of the fruits and the seeds. And the fact that this long time means that the plants are exposed to all sorts of, let's say, problems, dangers. This is a group of about 20 plants of 𝗀𝗵𝗶𝘀𝗀𝗱, where we went into the field and we found lots of flowers. Okay, when the plant is flowering, it's very nice. But then you come back after a month and a half, and the plants have been eaten or destroyed in some way. In this particular case, it was a group of plants where there were lots and lots of flowers, lots of fruits were produced, but this is the only single fruit that actually survived. Okay, so my point here is that there's a difference between seeing a group of plants in flower, maybe you see hundreds of flowers, and then what the population can actually produce in terms of seeds very often is much more limited, with only a few individuals that are actually able to go ahead and produce the seeds. So yeah, part of this is maximizing the number of fruits that can be produced. Or also, as has been said in the comments now, also protecting the plants can be an important method. The idea then is to get to this point where we have fruits and seeds. These are different species that we find in Lombardy. For example, this is 𝗵𝗵𝗶𝘀 palustris. I can see Dactyriza transdaineri, 𝗀𝗵𝗶𝘀𝗀𝗱𝗁, 𝗀𝗵𝗵𝗀𝗁 damasonium. Okay, one of the things that you notice is that I'm able to name the species looking at the fruits. Okay, this is actually quite a difficult thing because basically you have to have a lot of personal experience of the plants. Again, not just of the flowers, but you have to keep going back to them afterwards and checking the form, how it develops. Essentially, there's only one guidebook to identifying orchids from their fruits. It's also in German, so I don't personally understand it. But the point here is that you have to be certain of the identity of what you're collecting. And this can be very difficult especially for related species. Different species of 𝗵𝗵𝗶𝘀, for example, will produce 2 or 3 fruits which look very, very similar. The only way that you can be sure of the identity of what you're collecting is to go there beforehand and look at the flowers and make sure that you know the identity. So this is a big part of what we do. We have to keep going back and looking at precise points and making sure that we see the transition from flowers to fruits. This is just one example of how it can be difficult. We have 2 species of Gymnadenia that grow literally together. Okay, Gymnadenia conopsea, Gymnadenia odoratissima. Now, I can be certain, absolutely certain, that they're 2 different species because I saw them in flower and I came back afterwards. There are some clues. For example, we have some old flowers of Gymnadenia odoratissima here. But the only way that we can be absolutely certain is to follow the plants. So it's quite an intensive business of going into the field on different occasions and following the individual plants. Okay. When it comes to collecting seeds, this can be quite a problematic thing because each species will ripen in a different way. There are species which the fruits become dry and dark, and then they start to open and release the seeds. There are also species where the fruits will stay green, and they will even open when they're green and start releasing the seeds, and then you lose the seeds, but the fruits look as if they're immature. So one of the things that we found is looking at the color is not a very good indication of ripeness. The only way that you can be sure that a fruit is ripe is to squeeze it a little bit, to take it, to squeeze it end to end, just very gently. And if it's ripe, it will open. It has these ribs that run down the side of the fruit, and they open, and you'll see that some seeds come out. It's really the only way to understand if the fruit is mature or not. One of the other things that we found is that if we collect seed after it's rained, okay, so it rained in the morning, we go and collect the seed. If we sow those seeds, they will be full of fungi. The rain brings all sorts of spores and fungi and bacteria into the fruit, and it contaminates everything, and it simply becomes much more difficult to clean and then to use this material. So we never collect seed after the rain. So just a few other tips on how to collect seed. We usually use glass containers because we have fewer problems with electrostatic charges. If we use plastic containers, the seeds will stick to the walls of the container and it's very difficult to get them out. You can get them out, but you scrape a lot of plastic out of the container as well. So we're usually very sure to have some glass containers for collecting seeds. And we usually do the collection in situ, opening the fruits and collecting the seeds directly. You can collect fruits and open them later in the laboratory, but as soon as you collect fruits, or as soon as you have seeds in a container, you have to be very careful about the humidity. You have to let them dry out. Otherwise, they start to rot straight away. So you have to be very careful of the ventilation. So either collecting the seeds directly or collecting the fruits, you have to have a period where you leave them open in some way in the laboratory to dry them out. When it comes to seed storage, actually, I have to say that we tend to use our seeds straight away. The aim is to keep the seeds as fresh as possible and to use them as soon as we can. So we tend to store them, we dry them out for a couple of days and just on the bench in the laboratory, and we put them in the refrigerator, but the aim is to use them as soon as possible. You can, yeah, like 𝗂𝗵𝗶𝘀 is saying, you have to dry them out in paper. It helps if they're in a paper bag to dry them out properly. In the field, using glass is useful, but if you really want to get them dry, you can use paper. One thing I should add is not to use silica gel, because my colleagues at Kew informed me that it's too aggressive. It dries the seeds too much and they have problems germinating then. Seeds can be stored in the refrigerator for a couple of years. If they're dried out enough and they're at low temperature, they can be stored in the seeds, in the fridge. At the Millennium Seed Bank, this photograph here is actually at the Millennium Seed Bank in Kew. It's not particularly focused on orchids, but this is the structure where they store seeds at low temperature. They did a study in which they looked at the storage, the longer-term storage of different species of orchids at low temperature. And they found that different species reacted in different ways. They have a very strange storage behavior, in that some species can be chilled, some species can't be chilled, some can be chilled to very low temperatures, but not warmer temperatures. Some orchids can be stored at extremely low temperatures, but if you store them at higher temperatures, they die. And again, each species seems to be different. We don't really know why, but again, you have to kind of conduct your own experiments for your own species and try and understand what the conditions are for your particular species. I have to add to this that I have experience of people telling me that some species are very easy and other species are very hard to germinate and to store. And I found exactly the opposite pattern. And I suspect that it really depends on the local population and on factors like when the seeds were collected. And really, you have to conduct experiments for your own group of plants and your own seeds when it comes to storage. There is some information that seeds can be stored in liquid nitrogen, so at an extremely low temperature. Even tropical orchids can be stored at liquid nitrogen temperatures. I have to say that this is not something that we do in our laboratory for practical reasons. We can get liquid nitrogen. We can use liquid nitrogen in the laboratory, but using it every now and again and getting a small quantity is relatively easy. But if you're doing long-term storage over years, you need to have a dedicated facility, you need to have a dedicated project and money, and you have to have a continuous supply of liquid nitrogen. And personally, we don't have that in our laboratory, and we don't actually do this technique, but just so that you know, you can store orchid seed in liquid nitrogen temperatures. Okay. Okay, so we're running quite quickly through all of the various steps in producing orchids. We've looked at pollination, collecting seeds, storing the seeds. Now we'll look at germinating the seeds. And I'll be looking, of course, at the in vitro method, so in closed containers. And the advantage of this is that we can exclude other organisms, and we can give quite a specific growth medium, and there's no competition between the orchid seeds and other organisms such as fungi or bacteria, because they're excluded, because we do this in a sterile environment. Now, the idea is to control the environment in which the seeds are germinating. And one of the controls that we have is the availability of organic molecules like sugars that the plants will use for growth and germination, and also minerals, inorganic ions. They're usually metals such as boron or magnesium or manganese, which are used in the growth of the plants. So it gives us a great deal of control over the concentrations of these elements. And I think some of the general concepts we can see in this recipe. We don't need to look at the details of this recipe, but you'll notice that there's a whole list of amino acids. Now, there's a reason for that. This is probably one of the most important things for orchid germination, is that if you have high concentrations of inorganic nitrogen, such as nitrates or ammonia, this can actually be toxic for orchids. Many of them grow in sites where there are very low concentrations of inorganic nitrogen. And if you have too much in your growth medium, it's actually toxic. They won't grow. It will stop their germination. Orchids are adapted to getting organic sources of minerals and elements, because in nature they have this relationship with the symbiotic fungus, and they basically take their nitrogen from the nitrogen sources that are in living cells, in the fungal cells. And this means amino acids. So we have a list of amino acids, and actually there are some hormones and vitamins in here as well. But the point is that we give nitrogen in a form that the orchids are adapted to use. This is a very specific recipe. It was developed for tropical species 𝗵𝗵𝗀𝗀𝗀𝗃 and the hybrid between 𝗵𝗵𝗀𝗀𝗃 and 𝗃𝗃, which is 𝗃𝗃𝗃𝗃. And it's been adapted for use in 𝗃𝗏𝗃𝗃𝗃, which, I mean, we do have species of 𝗃𝗏𝗃𝗃 in Italy. So this is a kind of specific recipe for 𝗃𝗏𝗃𝗃 species. We do have general growth media. This is one of the most famous, Malmgren's terrestrial orchid medium, which is suitable for a range of different orchid species. This is the original citation for this recipe. There are a number of important things here. So firstly, it's important to have a source of energy. Again, it should be organic, an organic form. We add sucrose, because orchids get, well, they're not photosynthetic when they're germinating. They're basically just a ball of cells that are dividing, and they're still not green, and they need a source of carbon, and they get that from sucrose, from organic compounds. So it's very important to have that sucrose within the growth medium. As we'll see later, it's important then to keep moving our germinated plants between containers. This is called replating. And when we do replates, we have to add an extra quantity of sucrose to the growth medium. They need more, they're bigger plants, and they need more energy. The other thing that's very important here is that Malmgren, again, he wanted to add a source of amino acids. So then the orchids are using this organic source of nitrogen to grow. And because Svante Malmgren was a surgeon when he was working, he's still alive. When I say was, I mean when he worked as a surgeon. he had access to human nutritional supplements of amino acids, and he used these in his recipes. And it's actually something that is a bit difficult to get hold of. And we had to come up with a different way of adding amino acids to these recipes. And we came up with something that works. We tested this experimentally. If you use extract of yeast, but also peptone, which is a kind of meat extract, which has a lot of different amino acids in it, this is very effective in stimulating germination, in providing these amino acids. Okay. And the other thing here, coconut milk. Coconut milk, what's that about? Here we have to look at a completely different aspect of in vitro propagation, because I said that in vitro propagation is about having a great deal of control over the concentrations of each ingredient. But actually with orchids, it can be effective to lose a bit of control, to add something that we call complex organic media, or COM. And this simply means a mixture of organic material which adds energy and nitrogen and even hormones. Now, if we take the example of coconut milk, we know very well what's actually in coconut milk. This is a paper which analyzes the different compounds that are found in coconut milk. In fact, there's a big table here. I want to just go through this. It's full of sugar, it's full of different elements, okay? But the thing that's really interesting is coconut milk is a natural source of different amino acids. I mean, this is a big list of different amino acids. It's also a source of hormones that stimulate germination. Cytokinin hormones are factors that increase cell division, and they help plants to grow, they help seeds to germinate. And if we look specifically at the different cytokinins which are in coconut milk, we can see that there are different cytokinins, especially zeatin, and in fact the highest concentrations are different forms of zeatin. And this seems to be very effective in stimulating germination. Don't forget that coconuts are actually seeds, and for their own germination, they need energy, they need sucrose, they need nitrogen, they need hormones to stimulate the germination process, and all of those things are in coconut milk. So by using coconut milk or complex organic media, we're sacrificing a bit of control, and maybe it's kind of like using a shotgun, just shooting our orchids with something very powerful to get them going. So we can make these different mixtures and test them. Okay, so we've lost a bit of control, but this is still scientific, because the point is to record and quantify the effect that they have. So these are experiments for different species in which we've quantified the germination percentage, and we've looked at banana, coconut, and pineapple. Okay, BCP, this banana, coconut, and pineapple organic media. And we look at the effects, and we can see, for example, that for one particular species, using the coconut medium is much more effective. It's statistically an increase with respect to a control where we haven't added the coconut milk. Interestingly, we can see from this that even though banana extract is used in orchid propagation, we found that it actually completely inhibits the germination of these species. So we learned to avoid banana, absolutely. So again, we've lost a bit of control. We're adding this complex organic media, but the point is to really quantify the effects and make sure that we're stimulating germination and we're actually doing something positive for the plants. This is an example where we've compared against the control. We've compared the addition of coconut milk, banana powder, yeast extract, and then pure hormone addition. So for example, these are all hormones that stimulate germination in other species. For example, 𝗪𝗪𝗔𝗓 stimulated almost 50% germination for this particular species of orchids, 𝗧𝗵𝗵𝗀𝗁𝗧𝗵𝗂. And so here we do have a great deal of control. We know precisely the concentration of the hormone that we're adding and the effect that it has. So much of the science that we do is actually very simple. It's basically optimizing the germination technique by quantifying and comparing different substrates and looking at the effect on germination. There are some effects that are difficult to quantify, but they're definitely positive effects. This is some work that we've done recently on 𝗵𝗵𝗶𝘀 𝗶𝗵𝗂𝗃, in which we always had very, very high rates of germination. We had a mean of 94% germination for this species, even in the control without coconut milk, very high rates of germination. But we saw, this is just an observation, we saw that where we add coconut milk to the medium, in fact, you can see these blobs of white fat from the coconut milk, we have a greater degree of development of the plantlets. In fact, these are the only plantlets that then went ahead and produced green leaves and produced big plants. So coconut milk, it can have a positive effect, depending on the species, for germination, but also on the subsequent development. So we found this to be extremely useful. I didn't realize that the sound of this video, I'm sorry, I hope the sound is not too much, but anyway, once we have a growth medium, have a system for germinating these seeds, we have to actually get them to germinate. This involves sowing them in sterile conditions. Now, if we take seeds from a natural environment and we sow them onto a sterile medium, of course, they're going to be full of fungi and bacteria, spores. We have to clean the seeds. We have to clean the surface of the seeds. And what we're watching here is using a weak bleach solution. It's basically 5% bleach, with a little drop of detergent, just to make sure that that solution sticks to the surface of the seeds. We agitate, you can see with this moving hand, we agitate them for about 5 to 10 minutes. We're looking for a color change as well. When they start to go white, it becomes dangerous, because then the bleach is starting to affect what's happening within the seed, and it could kill the embryo. So this is basically cleaning the seed, but we're also breaking the outer integument of the seed, the tester, the outer covering, and this also helps to stimulate germination. In fact, what I'm saying here with these paragraphs is that the surface sterilization technique is not just about getting them into the sterile culture, it's also about weakening that seed coat, helping water to get in and helping them to germinate. Now, these are results from another laboratory where they looked in great detail at the seed coat of one particular orchid, 𝗀𝗵𝗶𝘀, but from what I understand, this is a general mechanism in orchids. There are two integuments. There's an outer integument, which is this kind of network that we see, which is more about kind of capturing the wind and helping with wind dispersal. But there's an inner integument, which can be, some people call it a carapace or a carapax. And this is more about protecting the embryo that we can see inside. So it's a layer of cells, and we can see that it's brown because the cell walls of these cells are impregnated with lignin. Lignin is the molecule which gives wood its woodiness, its strength, and it's also kind of waterproof and isolates the embryo that's inside. So orchid seeds can be very dark. They can be a kind of dark brown color for the presence of this essentially woody seed coat. And so what we're doing with the bleach is to help break that seed coat, to help the water get in, and to help stimulate the germination. Essentially, one of the observations that we've made, and in fact, when you look at orchid seeds, you'll see that there's a whole gradient from basically white seeds to black seeds. It depends on the species. Many species will have brown seeds, but essentially, this reflects the amount of lignin in the seed coat. If the seeds are heavily lignified, if they're very, very dark in color, it's very difficult to get them to germinate. And the reason for that is that you have to break the seed coat. And if you use very strong techniques to break the seed coat, you're also risking to break the embryo, to damage the embryo. And essentially, I'd have to say that there are species that are very easy to germinate because they have a very slightly lignified seed coat, and these are the species with lighter colored seeds, so 𝗺𝗵𝗶𝘀 𝗧𝗵𝗶𝘀𝗿, 𝗵𝗵𝗵·Hl 𝗂𝗵𝗂𝗂𝗂, and even 𝗃𝗃𝗃. But there are also species with very dark seeds, which are very difficult to germinate. And these are generally, I think, species from colder climates. So 𝗀𝗵𝗵𝗀𝗂𝗁𝗃, 𝗂𝗵𝗵𝗃, these do require very specific germination conditions. So I think this is a very general rule, I think. You do have to look at each species and find the conditions for each particular species. But in general, if the seeds are very dark, it's going to be more difficult. Okay. After you've sterilized the surface of the seeds, of course, they're covered in bleach, and you can't just use the seeds like that. The bleach will eventually kill everything. So what we can see here is the next step, which is washing the seeds with sterile water. So they're washed and they're sterile, and then they're sown onto the surface of the agar in the agar plates, as you can see here. In fact, I'll just zoom in for a little bit more detail. This is spreading the seeds out on the surface. Here, there's actually quite a large quantity of seeds being sown in this particular example, but the idea is to spread them out because we want to give them the resources that are in the substrate. We don't want them competing with each other. And the other idea is that we also want to see them clearly with a microscope so that we can see if they've germinated or not. So we spread them out so that there's no competition, or there's less competition for resources in the substrate, and also that we can see more clearly with a microscope and count the seeds that have germinated or not germinated. So this is a very important step in sowing the seeds. We do compare different substrates. So when we work in the laboratory, we tend to organize everything on a grid system. So each particular column or row represents a different treatment, and then different replicates within that treatment. So different substrates, different species. This is one particular experiment where we're comparing coconut milk, malt extract, banana extract, but also the specific hormones. So we've produced a whole range of different growth media, and we're comparing the germination on the different growth media. Generally, most species will germinate best in the dark, so we tend to wrap them up in aluminium and put them in the right temperature conditions in the growth chamber. There is light in the growth chamber because there are other plants, maybe. But most species will need the dark. Some species require light. Again, it depends on the species. There are species of orchids which will need light. 𝗵𝗵𝗃 palustris, for example, will require light, but other species not. Most species will require darkness. So the aim is to get them germinating. Germination generally takes between 2 or 6 months, depending on the species. It's very rare to have germination before 2 months, but way before the germination happens, we tend to get, let's say, well, we don't get much contamination in our laboratory, I have to say, with some pride, but sometimes we do get contamination, and they're generally these different kinds of contamination. Here you can see seeds where we can see fungal hyphae which are growing out from the seeds. And this means that the sterilization of the seeds, the surface sterilization of the seeds, was not enough. So we haven't killed the fungi which are hiding in the crevices or the holes on the surface of the seeds themselves. Another thing that we can see are yellow spots on the surface of the agar. This is a particular bacterium called Staphylococcus aureus, which is actually a bacterium which we have on our skin. So this represents bad technique by handling objects in the laboratory and contamination from our skin. When we see bacteria or fungi growing within the growth medium itself, not on the surface but within it, this is when the sterilization of the growth media hasn't occurred correctly. There's been a problem with the autoclave. Sometimes we see Okay, 𝗵𝗵𝗶𝘀 is saying that some anacanthus will germinate within 10 days. Okay, that's extraordinarily fast for orchids. The fastest we've had in our laboratory. No, actually, 𝗵𝗵𝗶𝘀 estivales does germinate very, very quickly as well on that time scale. That's right. Although generally we're speaking about several months. Okay, so here we see fungi that are growing in from one side, and this represents a problem with the container where there's a little hole and things are getting in from the side. And this horrible situation here is where somebody has coughed during the sowing process, and this is contamination from the air. Okay, so even if we work in a laminar flow sterile hood, if you cough, generally when you cough, your cough can travel up to 2 meters in distance. Even if you're in a laminar flow hood and you cough, you're going to contaminate things. Okay, so we have to be very careful in how we work. These are some of the problems that we see in the laboratory, but generally it goes okay. We don't have contamination, and what we hope to see is this situation where we have some ungerminated seeds, okay, but then we have germinated seeds. This is a body called a protocorm. It's basically a ball of cells that are dividing. Some of these cells will elongate. These are single cells called rhizoids, and these are essentially acting as if they're roots. They're going to get water and nutrients from the substrate. And, well, they're not roots exactly, because roots are organs made of different tissues. They're much bigger structures. But the concept is that of getting material from the surrounding environment. So this is what we want to see, the development of a protocorm, maybe with a leaf that's starting to develop on the upper surface of the protocorm. So lots and lots of protocorms, lots of rhizoids, and the leaf starting to grow and starting to become green. At this point, the plants have to be put into the light. So we take the aluminium foil off and we start them growing in the light. But before we talk about taking those plants ahead, I just want to say something about replating. It's very rare that you can keep these plants in the same container for months and months and months, because they run out of air. They respire. They need oxygen. They need the air to be changed, maybe even only once, but they do need to be opened or transferred onto fresh media. Also well, even sealed Petri dishes will also lose water. The film that we use, the parafilm that we use to seal these containers, does have some small holes in it which let water out very gradually over time. It's not completely sealed. And because we're talking about months, this long time scale, we do need to transfer the plants into new substrate periodically to make sure that they don't run out of resources and die. So it's an important part of it. Eventually they get too big, or there are too many of them, and we have this situation where they're becoming green. They have a leaf which is green, and there are too many of them in here, and they need to be transferred. What we've decided to do in our laboratory is to use these small tubes called DeWitt tubes. Sorry, that's still in Italian. This should be DeWitt tube. The idea here is that we work with small numbers of plants and we use lots of containers. This is quite expensive. This is the most expensive part of the work that we do. These containers cost a lot of money. But the idea is that if you produce hundreds of plants in one container and it's contaminated, then you're throwing away everything. And especially if you're working with very rare species, you might have enough seeds just to make 100 plants. So if you're using one big container and it gets contaminated, you throw everything away, like a whole year's worth of production thrown away in one night. If you use lots of little containers and some of them get contaminated, even for a rare species, you can guarantee that some will survive and you can produce the plant. So it's about giving us a security, a guarantee that we can produce these plants. So here we can see some plantlets of 𝗵𝗵𝗶𝘀. where we can see a single green leaf on some of these protoforms. These are actually getting a bit too big now. And the idea, there are definitely too many plants here. This is a dangerous situation of competition between all of these different plants. It gets very difficult to separate them when they have this much growth. But anyway, the idea is to transfer them into the new container. The medium that we use, the growth medium, is very similar to the original medium, but we add extra sugar, sucrose. We maybe double the sucrose concentration, and then transfer. It's taking a clump of the old substrate so we don't damage the rhizoids, and we transfer the whole thing in. Even if we take some of the old growth medium, the various organic compounds will still diffuse through the growth medium and reach. So that's how we do it. So then we have lots and lots of these DeWitt tubes which go into the growth chamber. Here we can see plants that are in the Petri dishes. So hundreds and hundreds of Petri dishes with different species germinating. And it's the production chain where we take them out of the Petri dishes and into the DeWitt tubes and keep going until we get to the greenhouse. So producing lots of green plants for all of these different species and keeping this process moving, this production chain moving, and getting the plants into the greenhouse. So these are just some of the productions that we've done over the years. Here, there are two examples of something very interesting. This is a key point. On the left, there are green plants, and they look okay. It looks like something positive, something that we would like, okay? And on the right, there are plants that look as if they're dead or in bad condition. But actually, this is what we want, because what's really important is the formation of a tuber underground, well, in the substrate. Once the plants have produced the tuber, they're safe. These tubers are extremely hard physically. They're very resistant. And basically, all of the work that we do to translocate the plants after this point has to be based on the tubers. One of the things we'll see in a bit is that if we move the green plants, they die, they're too delicate. But if we move the tubers, then we have a great deal of success in establishing these plants. So we actually work with the tubers. So the aim is to get these tubers. Depending on the species, especially the kind of species and the habitat, we have to mix up a substrate in the greenhouse. This is a particular mixture that we use for plants from xeric grasslands, so sandy soils. In fact, we add sand. And the point is to have a kind of heterogeneous mixture. We need micro zones where there's some water held onto the organic material, and we need zones where there's air, where the water passes through, where there are not many nutrients. These plants grow on nutrient-poor soil, so we have a mixture of sand and compost and perlite, so that we have particular micro points where there is water and nutrients, and others where the water is free to drain through, and it's a bit dry and there's a bit of air. We found that this works very well for large-scale production. One of the things that we do when we transfer the plants from in vitro is to keep them in humid conditions using the old in vitro containers. And we keep them there for about a week like this while they get used to their new conditions. And eventually we take these containers off and we start to get this is the growth of new leaves within this new situation. So at this point, we're really starting to cultivate the plants in the greenhouse. Here we put stones on the surface because we noticed that moss can get in here and it covers everything, especially with small orchids. Moss can be a real problem. So we put these little stones around the plants so that we keep the moss off. We found that this can be very useful. Again, we can see the moss invading and swamping everything. The culture method depends on the species. This is an example of a species from a swamp habitat, basically, which we actually found that keeping it with its roots in the water is very dangerous. The plants will die if their roots are directly in the water. I think the point is that these plants are just above the water in their natural habitat. Their roots are in the kind of peat and mossy layer just above the water. So again, the message is that you have to look at each particular species and the ecology of each species in order to cultivate it successfully. For example, woodland understory species like 𝗺𝗵𝗶𝘀𝘀𝗀𝗃, we found that a very good way of growing them was on a mixture of coconut fiber and broken leaves, leaf litter. And we found that we could cultivate these plants quite well. So again, you have to look at the species and the ecology of each species in order to cultivate it. So this isn't necessarily a good result in terms of conservation, because we're not really interested in getting the plants to flower in the greenhouse. But this was a nice moment. This was the first time for us that we got plants to flower in the greenhouse. So we were very proud of this moment. This was many, many years ago, probably about 14 years ago now. But again, the aim is not to produce plants that are flowering in the greenhouse. The aim is to get the plants into the wild. So I'm not necessarily interested in cultivating them up to this point. It's just that it is nice when it happens. Okay. So one of the things that we found about translocation, getting our plants and putting them back into the wild, we started off with these beautiful adult plants, taking the whole container, the soil, the roots. We did experiments with the whole thing, getting the whole ball of soil, or even washing off the soil and kind of putting the roots in and putting soil around and watering it. But the main message that we found from these experiments is that the plants are too delicate. If you do anything to these plants when they're in this stage of growth, you will break these roots. There are only a few roots, and you kill the plants. We found 0 success rate with these kind of translocations. Okay, what's really important again is the tuber. When the plant goes into a resting phase, usually this means during the summer, when the plant is actually losing its leaves and its roots, but you have a nice tuber, this is actually the best moment to do the translocation. I mean, these things are literally physically hard. I had tubers of some plants that I forgot about. I had them on my desk and I left them there for a year and they shriveled up. They became very shriveled and old and small. And I thought, well, I'll try and put them into some soil and see what happens. And they swelled up again and they started growing. And basically, once the plants are at this point, it is very difficult to kill them. So this is the point that you want to get to when you're transferring them. After the summer, they start to grow a new leaf and a new root, which we can just see here. And actually, I have to say, I'm sorry, but the arrow should be pointing here. There seems to have been a problem with moving the photo around, but this is the root that's starting to grow. And this is a good moment for translocation, even for small plants. Once they have the tuber and it's starting to grow, you can transplant them. And they should be put at a certain depth, more or less the depth that's equal to the size of the tuber. So this depth here is more or less equal to the depth of the tuber. This is a general rule when you're sowing seeds or tubers, that the bigger it is, you have to bury it slightly deeper, but not too deep, or the plants don't reach the surface. So this is the work of translocating, putting the plants back into the wild. In this particular case, it's a population reinforcement. One of the things that we found was very important, something that Thomas talked about before, was aftercare. And the aftercare that we do when we can is to put water directly after the translocation. It's very important to put water because essentially the plants are being disturbed and the soil isn't necessarily sticking to the roots. And if you put water on, it takes the soil and it sticks it onto the roots and keeps the roots humid. And it's very important for the survival. Aftercare, like Thomas said, is very important. I have to add, though, that very often our projects are to produce the plants and then we hand them off to a park or to somebody else who does the actual work of putting the plants into the ground, or doing the aftercare. And very often there is no aftercare. There's no financial resources for the aftercare. In practice, it's somebody else that's doing it, and you're not sure that they're doing it correctly. And you don't necessarily have the time or the resources to go and have a look regularly. So where we have our own populations in our local situations, I can go and check myself and I can do the aftercare, but I have to do it myself personally, because in these short-term projects, there's no money for that kind of work. And where the projects involve other parks and other people, very often I'm very unsure of the aftercare that goes into it. And in fact, I suspect that very often there's little aftercare. I did actually have one project where I handed the tubers and the plants to people that were putting the tubers down. And one guy had a hammer and he was kind of playing with a hammer and throwing the hammer up in the air, and it fell and it landed on the plants. And it's kind of one of these moments of disillusion when you realize that you've put 3 or 4 years of work into something, and then the person that takes it from you then just really doesn't care about it. So there are these kind of political complications to doing translocation and aftercare. But it is a method that works for particular species. We see the plants growing afterwards, we see them flowering afterwards, we see them producing fruit afterwards. One thing that I think Thomas said that's very important is that it's not enough to see them flowering. It's not enough to see them producing fruits. The aim is to produce a viable population. And what I have to say about the translocation studies that we've done is that I'm not sure if in 20 or 30 years' time that population will be viable, because I do see a slight decrease in the number of flowering individuals over time. And I suspect, again, that there are losses in the population, and I'm very unsure if we're actually able to establish a viable population. And with orchids, with the timescales involved, I think you really have to follow them over decades to be sure that there's been a real positive effect over time. So again, these projects, you're putting in a lot of time in producing the plants, but the outcomes can sometimes be very uncertain, I have to say. They can be very disillusioning, actually, sometimes. So I'd like to finish this talk now just with a few general pieces of advice if you want to do this kind of work. So I'll just list them very quickly here to finish. The first, and I think most important thing, is to do hand pollination, to leave some seeds in the wild, to have a minimal impact on the small populations of rare plants, and also then to collect some seeds that you can use in the laboratory. And this is a very valid conservation activity in its own right. There seems to be a difference between species, especially those from colder climates or colder situations, and Mediterranean species, where the colder species have much more rigid and difficult dormancy mechanisms, particularly linked to the seed coat. One method that you can use to get around this is to sow immature seed. I've not shown you this method, but you can take the seed just before it's finished maturing and sow that, and it does seem to work for some species. If you're starting out learning how to do this kind of method, it's very useful to use species such as 𝗁𝗶𝘀 because they're generally very easy. So you can, I think it's important to practice and to start learning on something that is going to work. You don't start learning using something that's almost impossible to germinate, because psychologically it's very difficult to keep going in this kind of work if you do months and months of work and it just doesn't work. You have to have something that works to get you used to the techniques and get you using the methods. Coconut milk, I'm just going to say that with one phrase: use coconut milk. Okay, coconut milk is your best friend. It's important to replate periodically the plants, otherwise they will use up the resources. I suspect that they will use up the oxygen in the containers as well. I think this is a big part of the plants becoming brown and dying. And then when they die, they actually produce toxic compounds that kill the other plants around them. So it's you have to replay to avoid this situation. Another thing is to never throw anything away, because I've found that even things that look dead will come back to life. Okay, so unless it's really contaminated, I try to keep everything, because you'll be surprised at what comes out 1 year later from the Petri dishes. Another thing is to be very mindful of the temperature regime, to think about what the plants will experience in the wild and to try to emulate this in the growth chamber. And the other thing, the other message that we've already seen, is only to transplant the tubers when they're resting, and not to try and transfer or move the green adult plants, because they're just too delicate. So this is basically the results of, it's kind of a synthesis of the work that we've done over the last almost 20 years or so. I think they're the most useful tips that I could give anyone that's trying to do this kind of work. So I'm aware that I've left out a lot of detail. For example, the immature seed, I didn't talk about that. But if you want to talk about these things in the questions, I'll stop sharing the screen now and we can have the question session.